Okay, so I'm Denise Thiede, and I'm with the Minnesota Department of Agriculture. And today I'm going to focus in more detail on the two pathways that we know a little bit about, and that's the seed pathway and the screenings pathway. And I'm going to try to highlight what I think are some of the things that we've done to be successful on the seed pathway in particular, but also what some of the risks are associated with it. All right, so as everyone knows, all seed that's sold in Minnesota must be labeled for sale. And there's a lot of testing that goes into accurately and truthfully labeling seed. This label shows you kind of a complex conservation mix. There's a lot of different species included in the mix. Every single one of those components would have been tested according to established methods for testing. And those tests include both germination tests, but also the physical purity. You know, what is the composition in that seed lot? What other crop and weed seeds are present? And it includes a noxious weed screen. So if you see on this label, the percentage of weed seeds, 0.01% noxious weeds, none found. So seed companies have a very well-established process for determining all of this information, and Minnesota has a regulatory program to monitor and make sure that labels are accurately portraying seed quality in the state. So one of the things that I think Minnesota did well is that before adding Palmer amaranth as a prohibited noxious weed seed, they made sure that a detection method was available for that species. So Ohio had actually added it as a prohibited noxious weed, but there was no way in seed testing to actually be able to identify it. So seed companies didn't have any tools available to actually detect Palmer amaranth. So we created a detection method before or during, while we added it to our prohibited noxious weed seed list. And this tool was essential in allowing seed companies to basically comply with this new requirement. So I'm going to just talk a little bit about why this tool was important. So you can see most seed testing is a visual analysis of seed. This happens to be a seed mix, and so the seed analyst is looking at every seed, identifying them to species. Unfortunately, for Palmer amaranth, it is not possible to distinguish that species from the other amaranth species. The seeds all look alike. So the only way to actually figure out if you have Palmer or not is to do genetic testing. So Minnesota sponsored the development of an ITS sequencing approach. It's a genetic test where you actually sequence a certain region of the ITS gene, and then you compare that sequence to a library of knowns to figure out if you have Palmer or not. Well, that original test was very expensive, over $100 per single seed. So you can imagine seed companies were pretty irate that we were going to start requiring that test. It did add some cost to their testing methodologies. Fortunately, while that test is still available in California at the California Department of Ag, there was a researcher at the University of Illinois who was doing work on this species and had identified a gene sequence that could be detected using PCR, which is a cheaper technology. He published a paper, and that technology then was adopted by 2 commercial labs who are now offering this service at $10 a seed or $30 to $50 for a pool of up to 100 seeds. So the costs came down considerably in terms of the detection methods. But this is the genetic testing that we are using in Minnesota, that we are requiring seed companies to use to make sure that they do not have Palmer amaranth in the seed that they're labeling for sale in the state. So one of the challenges is that seed testing is done, by commercial and internal labs throughout the U.S., and not all seed analysts have the same technical expertise in terms of visual identification. Not all labs are following the recommendation of genetic testing. So, in fact, one of our Minnesota seed companies did a survey. He sent out samples of Dakota switchgrass to, I think, 10 different seed testing labs. He knew that the seed had Amaranth in as a contaminant. It wasn't Palmer, it was a different pigweed, but he knew it was present. And of all of those labs, only one offered genetic testing as a follow-up test. So seed testing labs are not necessarily adopting this new approach, and they're not integrating it into their seed testing protocols. And so one of the steps that we're taking is we are adding a statement. So seed analysts consult an all states noxious weed seed list. When they're doing that visual exam, this is the list that they use to determine what is a prohibited noxious weed seed. And so for Minnesota, we're going to add in that list that genetic testing is required to help drive that change in the seed testing labs to make sure that they're offering that as a tool and reporting it correctly on their seed testing reports. So in Minnesota, as I mentioned, we have a very robust regulatory program where we actually pull samples of seed across the state and we monitor the accuracy of those seed labels. And so you can see for the last 10 years, we actually have a pretty high compliance with the Minnesota seed law for all the samples, over 90% compliance. Because of this Palmer Amaranth situation, we wanted to increase our surveillance to make sure that we were finding companies that weren't doing the genetic testing as required and were introducing this weed seed into the state. So what we did in 2017 is we increased the number of native seed samples. That's the first pathway that we had identified coming into Minnesota in 2016. We decided that we needed to focus on that. So we increased from about 20 samples a year to 80 samples in 2017. And last year we collected 138 native seed samples. And we're monitoring those samples to see if Palmer Amaranth is a contaminant. In 2018, we had 3 of those seed lots contain Palmer. 2 of them had come out of Kansas, and 1 of them is of unknown origin. So it's a small number of seed lots, but that is what we're trying to catch. We need the companies that are selling and labeling that seed, we need to make sure that they are following the appropriate testing methods. Okay, so doing these investigations is complicated, and Tony talked a little bit about this, about what's happening in Minnesota. And what I'll focus on the 4 counties in northwestern Minnesota, trying to determine how can we identify what is the source of the Palmer Amaranth coming in. So in that case, that happened in 2018, January 2018. We detected a seed lot that was for sale, and that seed lot had been sold to landowners on 1,400 acres in those 14 counties. So we knew that the seed contained Palmer Amaranth, but we didn't know if the plant was on the ground. So in 2018, we actually went out and surveyed all of those sites to determine if Palmer was present on the property or not. So fortunately, we had been able to conduct that noxious exam on that sample. We detected Palmer, but we could not get the records for the seed lot. So that seed lot, the labeler did not have the records that are required by law for tracking back where did that seed come from, who had they purchased it from. And so basically we were stalled. We couldn't get any further back on that sample. And so that's one of the things that we're finding is that unfortunately, in many of these cases, the records that are required for selling seed in Minnesota are not being maintained. And our seed regulatory team this year is actually going to start doing records inspections at seed producers to start trying to identify where we're seeing a lack of compliance in terms of record keeping. So that'll be a focus for us. One of the other things we learned is that seed companies, because in the past the noxious weed seed list did not change that frequently, they would only do the noxious exam once, and then that seed lot might carry over multiple years. So between 2016 and 2017, we added Palmer Amaranth, but some people were still using the noxious exams from 2016 when Palmer wasn't on the list. So just educating people that you may not be able to carry over those noxious exams on seed that you're labeling for sale. So what we learned, and Tony has touched on this briefly, but you know, if you look at Lyon and Yellow Medicine counties down here in southwestern Minnesota, we had one seed mix that was planted in those 33 sites. It was confirmed, Palmer was confirmed in the mix, but we could not identify the source component, and again, why not? Because the vendor did not have required records. The same thing in 2017 in Todd and Douglas counties in the center of the state. We confiscated 10 seed sources from one of the landowners in those conservation plantings. None of that seed was labeled. When we tested it, we found a grass mix that contained Palmer. Again, we could not get the required records to track back where that seed came from. The risk is, where else was that seed sold in Minnesota? If we had the records, we could figure that out, but without them, basically our hands are tied. So those, you know, we are taking enforcement action. Actually, in all of these cases, it's all still pending, but you know, enforcement and deterrence is going to be a key part of getting people to comply with this new requirement. So in 2018, you know, we found this contaminated seed source. Again, seed records and file samples not available. All right, so now I want to talk a little bit about screenings. So Minnesota has a Screenings Act, and so we have some statutory authority to inspect screenings. These screenings are basically chaff and debris that is generated when seed or grain is cleaned, and this act covers the use of those screenings as feed. So these screenings either have to be less than 10% weed seed or contain less than 90 seeds per pound of restricted weeds, or the screenings have to be devitalized before they're used as feed. And so we decided, based on the find in Redwood County, that we needed to go out and do some screenings inspections just to understand what's happening on the screenings pathway in Minnesota. Really, you know, we haven't really had any regulatory oversight on this act for the last 20 years, and it's really related to the widespread use of herbicides. You know, there just hasn't been a lot of weed seed in the seed that's being cleaned or the grain that's being cleaned. So we went out and we took a look. We've collected 9 samples so far, including sunflower hulls, wheat screenings, some bird seed screenings. The number of amaranth seeds, on average about 522 seeds per sample. So there's a lot of pigweed seeds in these screenings, a tremendous number. One of them had over 2,000. We did genetic testing on all of these amaranth seeds, and we found, Tony stole my thunder here, but we actually think we found the source for the Redwood County plants. That the sunflower hulls were being fed to cattle in a feedlot, and the manure from that feedlot was being sold to that landowner. We were able to go to one of those feedlots, sample the sunflower hulls there, and we did detect Palmer amaranth in those sunflower hulls. The rate of the Palmer amaranth in those screenings was really low, and that's consistent with what we saw in the field. We only had 1 or 2 plants in the field. But this is a new and important pathway that we're going to need to start monitoring in Minnesota and figure out how we're going to prevent Palmer from moving in this pathway. All right, so I think we've had a lot of good success on the seed pathway. I think seed companies are implementing methods to make sure that they limit Palmer amaranth in their seed production fields. There's a lot of tools and techniques that they can use to do that. Companies have embraced these new seed testing practices that include genetic testing. And one of the things I wanted to point out is, Wisconsin has not only added Palmer amaranth, but it's also added water hemp as a prohibited noxious weed seed. And so one of the things that they will need to use genetic testing also for water hemp. Both commercial labs, Eurofins Biotechnology and the University of Illinois, have both validated the water hemp assays. So this assay is available on the same DNA that they're using to test for Palmer. So it should be a smooth transition. In Minnesota, we didn't add water hemp, so it wouldn't be required here, but it would be required for sale of seed in Wisconsin. And we'll continue to have regulatory oversight, including using enforcement and penalties as a deterrent to companies that are not following the requirements of the law. One of the things that Dan Shaw is going to talk a little bit about next is just how important cooperation has been within state and federal agencies in Minnesota. Kind of like Jeff's triangle of trust. We've really worked hard to work together to figure out how we can all address and tackle this problem. And I think that not blaming one agency, but trying to work together to say, hey, we're all in this together. We need each other. We need to all, take the best step forward. And I think cooperation is a key to success within the state and also at a regional level. So, thank you.